Hi, I'm Chris Olson, and this is my project, Engineering Stealthy Exosomes for Drug Delivery, under my advisor, Dr. Bill Liu. Special thanks to Mika Domingo as my senior design partner for this project. So why do we want to engineer exosomes? Exosomes have great potential as a drug delivery system because of their low immunogenicity and high biocompatibility. In addition, exosomes have natural roles in cell communication, making them an even better candidate for drug delivery and drug loading. Various kinds of drugs, such as small molecule drugs, DNA, RNA, and proteins, can be loaded into exosomes, and then exosomes can then be injected into patients to deliver that cargo to therapeutic sites. My project seeks to improve the capability of exosomes as a drug delivery system by giving them additional immunosuppressive factors. I seek to engineer exosomes to avoid the main mechanism of immune attack for exosomes, that is macrophage phagocytosis, by including CD24 or CD47 proteins don't eat me signals that will turn off phagocytosis when interacting with macrophages. I designed 4 different plasmids to test the success of our experiment. 2 plasmids of my unmodified protein with a GFP signal for analysis and verification, and 2 proteins that included a further transmembrane localization signal, TVSVG, to improve membrane localization. This is important because in order for our protein to be involved in exosome production, they have to first be present on the cellular membrane. My project was broken down into 3 main steps. First, I did a transfection as a proof of concept to ensure the viability of my experiment. Second, I did an exosome harvest where we tested the design and actually harvested exosomes to test and analyze. And finally, we did an analysis of our harvested exosomes to determine the success of our experiment. First, looking at a transfection, we saw that the constructs were localized differently in the body. We saw, or in cells. We saw that good membrane localization for CD47 constructs and poor membrane localization for CD24 constructs. This means we expect CD47 constructs to produce more modified exosomes than CD24 constructs. However, we did see CD24 constructs had intracellular membrane localization, which still could produce exosomes as that's part of the exosome pathway. Moving forward to our actual exosome harvest, we saw that a lot of our initial ideas were confirmed. A CD47 TVSVG showed the best membrane localization and the best exosome production overall, and improved production over CD47. However, it's less clear whether CD24 TVSVG improved membrane localization and exosome production over CD24. Overall, we saw that CD24 performed poor compared to CD47 constructs, as CD47 constructs had superior membrane localization. We also saw all of our samples produced some exosomes, with CD47 TVSVG showing the best exosome targeting overall and the best success for our experiment. Going forward, we would like to further validate our experiment by characterizing our exosomes further. Exosomes are known to be very heterogeneous, and so characterization techniques such as nanoparticle tracking analysis, proteomics, and NGS RNA sequencing would allow us to determine more about our exosomes. Nanoparticle tracking analysis would tell us the count and size of our exosomes to determine what population we have. Proteomics and NGS RNA sequencing would tell us what exactly is in and on our exosomes, which would be important for medical applications, as we would have to prove that our exosomes would be safe for the patient. Overall, we showed a good proof of concept to show that exosomes can be modified with additional immunosuppressive factors, and we will now look forward to determining the exact composition and success of our experiment.